Thanks very much. It's a pleasure to be here today. What I'm going to do, all of us work together, by the way, on research projects dealing with stormwater, pesticides, other pollutants, and salmon and other species, aquatic organisms primarily. What I'm going to do today, since I'm the director of the WSU Puyallup Research and Extension Center, is tell you a little bit about our programs, what we're doing, and what direction we're moving in. This center is located about a 10-minute drive up Pioneer Avenue from the Tacoma Dome. So it's very close, and if you like one of these days, come by and we'll give you a tour of the center. Okay, this center was established in 1894. It's basically was an ag experiment station, and as time has gone on, the mission has changed. The original mission was for hops, working on hops research, because the Puyallup region was the hop capital of the United States. A lot of people don't know that. The hops industry now is located in eastern Washington, and that's because an invasive species showed up and destroyed the crop year in and year out, and WSU scientists got clean stock in a greenhouse and moved the whole industry to the other side of the state where it resides today. But as time has gone on, this area has urbanized greatly, and we've had to redirect the center, and we've done that and now work in environmental and human health issues. We have a large human health program. We have a nursing program. But anyway, the center has about 125 people. We have about 20 faculty members, including engineers, agriculturalists, toxicologists, and a whole slew of others. And we have graduate education there, where we have quite a large number of graduate students who pursue both masters and PhDs. I'm going to talk to you a lot today about our Green Stormwater Infrastructure Research and Education Program. The educational program is run by Curtis Hinman. I'm not going to go into a whole lot of detail about that, but he does a certification program and educates stakeholders that are involved in stormwater permits primarily. We have a very large low-impact development or green stormwater infrastructure program at our center. This was funded by the Department of Ecology along with money from our center. And we had many sponsors and groups that helped us and worked with us, especially the city of Puyallup. And the goals were to evaluate various green stormwater infrastructure techniques for their ability to reduce stormwater volume and pollutant loads. And some of the participants in this are Tanya Lee Irwin, who I'll mention later, as who's the manager of the Washington Stormwater Center, Curtis Hinman, I mentioned already, Nat Scholz, of course, Jen McIntyre, and Jay Davis from U.S. Fish and Wildlife. I want to show you some pictures of what this looks like. We have a parking lot, our main parking lot, which now consists of pervious asphalt and pervious concrete. But unlike a regular parking lot, these are lined cells where we can evaluate these technologies for their ability to reduce flow and remove pollutants, and it's replicated and scientifically defensible. So off the back end of this is what this looks like, where we collect stormwater. We can do both natural storms and put on artificial storms, where we dose stormwater and put it on there so we know exactly what pollutants are in there and how effective these systems are. Some of these cells are maintained with vacuum street sweepers. Others are not maintained because we're trying to figure out when they fail. And some of them are just regular pavement as a control, in other words, not pervious. And then again, we have the same kind of a situation for concrete, and we're also looking at wear and tear and how these systems hold up. Along with this, we have rain gardens or bioretention systems, and we have a series of these. This system here, again, these are lined cells, plumbed like the other system we showed you. Some have no plants, some have only grasses, some have shrubs and grasses, and some have just shrubs. The point here is they all have the same soil mix, which is the standard 60% sand, 40% compost that the state recommends, but we're trying to figure out whether plants have an influence or what influence they have on pollutant load removal and flow reduction. We have a series of scientists working on this, hydrologic engineers, horticulturalists, turf experts, and as you'll see, toxicologists as well. We also have another series of rain gardens here. These all have the same plant mixture, a standard plant mixture, and here we're looking at a whole series of different soil amendments and different types of mixtures of soils. And the point of this is to determine whether we can tweak these systems and make them more efficient at removing specific types of pollutants from stormwater. Another thing that we have are a series of columns which are also bioretention systems. We have different size columns, but we have these extremely large columns that are pictured here, which have both plants and no plants. And again, we can put different soil mixes in here as well and test those for their efficacy for removement of pollutants. And then tied with all of this is the WSU NOAA Salmon Lab, which Nat Scholz and I developed about, what, 10 years ago, Nat? And the thing that's interesting about this is we've been working on mixture of pollutant mixtures on salmon health for a long time. And then when stormwater became a big issue for us at our center, we've now tied this lab into that work. And Jen's going to talk about that a bit in a few minutes. But more importantly is the fact that this is a very large building. We're only using 1/3 of it. And through the Economic Development Board's Clean Water Technology IPZ, we received a large sum of money to renovate that whole building and expand it. And so we're going to have a lot more than just salmon in here eventually, probably other fish species, other aquatic invertebrates, and perhaps shellfish. What I didn't mention too is we have an aquatic invertebrate lab too at our center, and Jen will show you some of that work as well. We also partnered with the Urban Waters Center at UW-Tacoma and developed the Washington Stormwater Center. Tanya Lee Irwin is the manager of that center. Kurt Marks is a member of that center. He's here today. Lisa Rosman is in the back, and others. And this is a resource for the state. It was actually a House bill that was passed into law, and it's a nonprofit joint venture between WSU and UW-Tacoma. And what we do in this center is a series of things. We provide education for stormwater permit holders, both municipal and business, and then, whoops, sorry about that. We also have the TAPE program, which is this emerging technologies program that Ecology used to administer. That's being run primarily by Kurt. And then we have a research program, as I've mentioned before, and so that's moving ahead very well. And that's funded by the Department of Ecology. Another thing we do at our center, Clark's Creek is a very important stream for the Puyallup tribe. It's got 3 salmon species that run through that creek. As I say, it runs right through our property. There's a Clark's Creek initiative. This is an award-winning program for the restoration of a creek that's got endangered species in it. We're working on the restoration of that and have been very successful so far. And then a final thing that we're doing is we have a STEM education program with K through 12 through the Puyallup School District, where we educate high school students on our property with Clark's Creek and various ponds and wetlands we have throughout our property. And then they train younger students. They act as docents, and that's been a very successful project. Now, along with this, to do these things, we have a lot of partners and a lot of funders, and we couldn't do this without them, so I want to mention a few of those. The funders are the Russell Family Foundation, the Boeing Foundation, the Bullitt Foundation. The Tacoma-Pierce County Economic Development Board has been a big supporter and helped us with this money for the renovation of the Salmon Lab. And then we have a whole lot of other partners as well, including various cities like the City of Puyallup, Tacoma, Seattle, Urban Waters, NOAA, U.S. Fish and Wildlife are partners with us. Various business organizations, the Department of Ecology, and of course EPA has funded a lot of what we're doing. So I want to let you all know that we're doing this. We're expanding what we're doing. We're growing these programs. We're going to have this really great capability in that toxicology laboratory that now is basically salmon and some aquatic invertebrates, where we're going to have other species eventually there to do work with. We are always looking for new partners, and so with that, I'll leave it to Nat. Thanks.
But I'm going to be here for the day, and so if you miss anything or have follow-up questions, just let me know. Most of the stuff I'm going to show is published, and I can share those publications as well. I am from the Northwest Fisheries Science Center in Seattle. We are the research headquarters for NOAA Fisheries. Stormwater, of course, is a huge issue for us. We're part of a large collaborative group that involves researchers, NOAA Fisheries, Washington State University, U.S. Fish and Wildlife Service, and then a bunch of project partners. And a lot of our funded work is intended to advance the Puget Sound Partnership aims for Puget Sound. Basically, the core areas that we're focused on for stormwater and non-point source toxic runoff. What are the issues? How can they be effectively minimized? And our ongoing efforts to reduce impacts working. And this talk today is only going to focus on the first bullet. This is a CSO about a block away from our science facility in Seattle. This is last October. last November, sorry. And again, delineating between the pollution that you see and the pollution that you don't see. So from a non-point source water quality perspective, this is really where we focus a lot of our efforts, trying to get out ahead of many of the salmon habitat problems that are chemical in nature, both for familiar classes of contaminants and legacy contaminants, but also increasingly we're focusing on emerging classes of pharmaceuticals and other contaminants of emerging concern. For several reasons, we've been focusing on coho salmon as a stormwater sentinel. The reasons for that is that they're widely distributed in Puget Sound. They like lowland streams that are relatively small. They tend to overwinter, so they're in freshwater for longer compared to pink salmon and chum. They're supported by a diverse food web, which is important from an ecological integration perspective. As I'll show you, they're very sensitive to water quality. And for NOAA, they are a species of concern. Again, when it comes to stormwater, most of our science is focused on freshwater life history stages, and we have done research over the past decade on embryos, on freshwater juveniles, and on adults. And I'm only going to focus today on some of our work on adults. That's shown here. So in the 1990s, the city of Seattle and others invested quite a bit of money in urban stream restoration projects. Most of it focused on physical habitat. And this is a video from 2000. One of the key things from this effort was essentially coming in after the projects were done and evaluating the extent to which salmon were using the newly available habitat. And we at NOAA started receiving widespread reports from around the region of coho salmon coming in the fall when it rained and water levels were coming up in streams and showing this suite of symptoms. So this is a freshwater entry adult coho female in Piper's Creek in northwest Seattle, and loss of equilibrium, loss of orientation, gaping, and the fish soon after died. And this phenomena we've called coho pre-spawn mortality. Our group and others have been studying this for more than 10 years at multiple urban stream sites in and around Seattle. And what we have found, I'm just going to go through this quickly, is that mortality among coho spawners has been widespread and has been high. These are, the way we quantify this is identifying adult females that are still full of eggs, these carcasses in streams. We had a big push last fall, so I'm just going to show some data from last year as well. The other thing that we've seen is that the symptomology is also quite consistent across years and across different streams, same progression. So loss of equilibrium, loss of orientation, pectoral fin splaying, gaping. You can see that fish at the top is surface swimming. We published 3 papers on this recently. One is a forensic analysis, and the major findings were that we're seeing adult spawners dying every year in urban streams. The phenomena is widespread. We think it's been going on for a long time. The rates are high, 60 to 90% of the total run from daily surveys. And the forensic sort of weight of evidence approach is pointing to toxic runoff being causal. The second study is a land use, land cover analysis. And so we did a big GIS effort to essentially develop predictive hotspots for coho pre-spawn mortality in the 4 central Puget Sound counties. And basically the land use analysis found a very high correlation with the severity of the fish kills and the amount of impervious surfaces within a watershed. And it also indicated that coho are likely to be impacted across large geographic areas. We have since gone on to validate these model predictions and are coming through with another generation of models right now. The third study was a population analysis, basically asking, with the rates that we've been seeing in Puget Sound urban streams, what are the potential consequences for coho that are in otherwise healthy, less developed watersheds over time with population growth throughout the region. And so what we're seeing from the modeling is that we're predicting rapid local extinctions at the 60 to 90% rate, which is not surprising. And the other thing, through population structure of coho salmon and metapopulation dynamics, that the mortality may drag down coho abundance in non-urban watersheds as a consequence of straying. We are now following this up with additional studies. As a matter of fact, we're waiting for it to rain. We'll be working with adult coho this fall. But the question we asked last year is exposure to urban runoff sufficient to cause coho pre-spawn mortality? We did this in collaboration with the Squamish tribe. So we're at the Grovers Creek hatchery facility. It basically involved collecting urban runoff and trucking it out to the salmon hatchery. And these are adult coho that are recently freshwater entry from the Sound, and they're in these long PVC tubes. You can probably tell which one's clean and which one's stormwater. And I'm only going to show you some of the qualitative results. We've collected essentially a ton of tissues. We're doing a genetic analysis that'll be ongoing this year, more forensic analyses related to this. But basically what we found is normal clean water on the left, the adult coho exposed to stormwater on the right. We see the same suite of symptoms. We did this last fall over 4 major storm events. Every single storm event basically killed the adult coho. So across these, and these are logistically difficult studies to do because the fish are big and they're not particularly abundant. But we found in unexposed salmon 100% survival. The fish that are exposed to stormwater, we had 100% symptomatic and dead fish within less than 4 hours. So we now have forensic tissues from samples that were collected in the field from Longfellow Creek in West Seattle. We have fish that were exposed under controlled conditions to stormwater and reference fish as well. And so that will be the focus for our genetic analyses this coming year. And that's just our website. So if you, we just updated the content on our website. So if you're interested, that's where you can find more of a background. And you can always email me for copies of the papers. That's it.
All right, see, I do juggling microphone and clicker. So I'm going to focus on the biological effectiveness side, and this is, as John mentioned, a project that we're all working on together. So the reason that stormwater is a problem for aquatic wildlife ends up being because this is not a forest. And like Ben mentioned in his talk, the reason this isn't a forest is it doesn't have the pervious surfaces, and I liked how he said it, that allows the water to slow down, spread out, and then soak in. So this doesn't act like a forest when it rains. And the goal of stormwater, green stormwater infrastructure, is to try to mimic the pre-development landscape, basically, so that surfaces that look more developed can maybe act less developed. And the idea there ultimately being that green stormwater infrastructure will end up being less harmful to aquatic wildlife. So the project I'm going to describe today is a joint effort. Both of these guys have mentioned it's hugely collaborative. These are some of the partners involved. The first part of the project looked at just pretty simply characterizing stormwater. We want to, if we're going to look at effectiveness, we want to start with something that's kind of nasty to start with, right? Otherwise, it's harder to tell if you've cleaned it up. So we're going to characterize the stormwater and evaluate just the baseline toxicity to some aquatic wildlife. So we went out there and found some pretty nasty runoff. So highway runoff around the world contains a pretty typical suite of a complex mixture of contaminants. So we had access to runoff coming off of a pretty busy urban highway. Here's what that looks like. Obviously very dark, very similar to what Nat showed with the adult salmon exposure. So some of the tools that we used when we first started characterizing the toxicity are an aquatic invertebrate, which is Cereodaphnia dubia, and there we're looking at a pretty traditional toxicity approach. We're looking at lethality and we're looking at reproductive impairment. And I'll show a little bit also of the research we've been doing characterizing stormwater toxicity using zebrafish, which are a very useful model aquatic animal, often used in human health because they're toxicologically, the response is very similar to humans. So I've got some interesting slides for that, and that's more looking at maybe trying to get at what some of the mechanisms of toxicity are. All right, so we've done this characterization work for 6 storms now, and I'm showing across the bottom just the month in which that stormwater was collected, and then showing the different concentrations of metals and also total PAHs for the different storms that we characterized. And the storm in red is the one that I will show at the end, where we actually, for the first time, looked at a biological effectiveness of low-impact development. But here I just want to highlight the biggest actors in the stormwater runoff as far as metals are zinc and copper. which won't be a surprise to anybody here. And then the PAHs was a more interesting story for us. Here I'm showing somewhat of an abbreviated fingerprint for PAHs, and these start from low molecular weight PAHs like naphthalene, and then move up to higher molecular weight, more benzene rings, things like pyrene, and then up here things like benzoapyrene, the human carcinogen of concern. And what I want to highlight here is that the biggest contributors as far as, now this is just proportion, but as far as concentrations were phenanthrene and also chrysene. So this becomes important a little bit later on. All right, so characterizing the survival and the reproduction for the invertebrate lab rat, we have at the top panel here for our different storms relative survival from a typical 48-hour toxicity test. And what we're seeing is that some of the storms were acutely lethal, some were 100% lethal, some just showed significant mortality. And in this bottom slide, we're showing now a 7-day test, which is a typical test for looking at more chronic exposure, and what it tells us is whether that exposure results in reproductive impairment. So we're seeing further mortality at the end of that period, and also reproductive impairment in this invertebrate species. Now, we also saw lethality for our aquatic fish species. Well, that's redundant, but for the fish that we used in these tests. But more of interest were the sublethal effects that we saw, and I'm going to highlight some of those here. So on the top left is an image of a control zebrafish larva at about 4 days old. And then the middle and right panels are zebrafish larvae for two different storm events, and these are just showing some very characteristic effects. First of all, we consistently saw an inability to hatch or a delay to hatch for these fish, and they end up hatching about 2 days old. Developmental delays. The asterisk is to denote a small eye phenotype, microphthalmia, which is related to impairment of the developing nervous system. Pericardial edema is sort of it's the bulb that you're seeing around the heart. So the heart's located between the yolk sac and the head, and there's a fluid accumulation that occurs there, and this is usually an indicator of a physiological problem with the heart itself, with circulation, although sometimes with the kidneys. And finally, deformed jaws and hearts were also very common. Now I have a few videos. I'll try to see if I can get those to work here. Just as examples, here is a control fish, and you can't see the 2 chambers of its heart, but you might be able to see the beating of those 2 chambers. This is a normally developed fish. And then here I've got 2 examples of some of the typical problems you'll see. Here you see 2 chambers of a heart beating, but there's no movement of blood through the fish's body. And there's also, difficult to see in this lighting, but cranial hemorrhaging, so blood spots in the head region. And then finally, where's my cursor? Anyone see my cursor? Hello? All right, well, this fish is really messed up. So he's got huge pericardial edema, this huge bulbous, the deformed jaw, the small eye, and also difficult again in this lighting, but you can definitely see the really misformed heart and the lack of red blood cells moving through it. Okay. I might have to pop out of this. Shoot. Looks like it froze on me. Sorry about that. All right, I can fix this. I can. You know, if anyone has any suggestions, though, I'm not against that. I might have to exit the program and get back in. No? He's saying no.
Hey, Jen, while he's doing that,
Yeah.
maybe we can have a question or two.
Please.
Either for you or for the 2 other speakers so far. We're heading toward that as well in any event. So any questions?
First flush runoff. So we were concerned that we might not be seeing the most acute responses if we looked at an integrated approach for collecting stormwater runoff. Okay. Thanks for that question. Let's move right along. I also want to talk this one slide about some of the tests we did looking at dilutions of runoff. So this is showing dilutions for one of the sublethal storms that didn't cause acute mortality for the fish. And these R's at the bottom are dilution, so R100 is undiluted runoff, and then it goes down in this slide to R5, which was just 5% runoff. And top down, we're looking at proportion of the fish that were hatched at those different dilutions at the end of the test period. So overall, what we're seeing is that the most sensitive effect was this effect on hatching, also swim bladder inflation. The next most sensitive was this impact on the eye area, down to a 10% dilution of a not very toxic storm event. So these are some of the sublethal mechanistic things that we're interested in. All right, as far as what's causing these impacts, we haven't gone very far into this yet, but typically the types of contaminants that are present in urban stormwater runoff, particularly highway runoff, are coming from these major sources. And studies that have tried to identify what the toxic components are tend to point the finger at metals, at oils and greases, PAHs typically, sometimes surfactants, and then in areas of the country where de-icers are used, those can obviously contribute toxicity. We've done a little bit of research to look at the bioavailability of metals in the runoff, because that was a big concern of ours based on our previous research with metals. And we have a really cool tool using zebrafish where you can expose them to a dye, and it will highlight neuromass in the fish. And so these are locations where there are peripheral sensory neurons, the ones that detect motion in the water. It allows them to tell about a predator lunge at them, about the current in the water. And they show up under a fluorescent light as these little glowing balls. If you zoom in on each one of those, what they actually are a group of hair cells, so little filaments stuck together, sticking out of the fish. And that's what you see in this picture, is looking top down at those hair cells. And in the top image, this is a control fish. And then if you expose that fish to a more higher concentrations of a neurotoxic agent, basically that fluorescent staining disappears. This example is fish exposed to dissolved copper. So we did this with our zebrafish, exposed them to undiluted stormwater runoff, and what we counted the number of those hair cells, and a value of 1 here means no different from the control fish. And basically that's what we saw for each of these storm events, was that there was no neurotoxicity. And this was surprising because the dissolved copper, which was the most neurotoxic component of the stormwater, to our knowledge, were actually quite high in concentration. However, dissolved organic carbon is known to sequester those metals and make them no longer bioavailable to the sensory system, to the neurons. And the DOC was also quite high in these samples, so it looks like DOC was protective for these samples of runoff. PAHs, on the other hand, are also quite common in stormwater runoff. Again, here's that fingerprint from low to high molecular weight, and a very consistent pattern among storms. Even though each storm was different, the distribution of these PAHs was remarkably consistent. These particular PAHs are known to be neurotoxic to animals. Fluoranthine, dibenzothiopine, phenanthrene, pyrene, fluorine, and then some of the higher molecular weight PAHs. And very interestingly, some new research that was done this summer at NOAA Fisheries suggests that there could be a synergistic toxicity between phenanthrene and chrysene, which was that other highest, most dominant pH in stormwater samples, which by itself is not cardiotoxic, but there may be synergistic toxicity between those two. So we have a lot more research looking at the potential for pH to be playing a role in this toxicity, but I don't have time to describe that today. So I'm going to jump ahead to the effectiveness part, which is the happy news at the end of the sad story. So this work took place at WSU in those large columns that John described earlier. We had those 4 treatments, and then we looked at a variety of toxicity tests using the invertebrates and the zebrafish that I described, but also 2 of our target species, juvenile coho and a wild mayfly nymph. So here's us with some of our collected runoff. We had to, unfortunately, mix it in this large plastic tank, which had its own issues. So in fact, this is sort of a conservative look at what was really going on. We took a subsample before we mixed it together just to know what we'd perhaps lost in that big plastic tank. After it's all mixed together, we took off enough runoff to have an untreated sample. And the rest of it we passed through the columns. And the columns, half of them had plants established in them and the other half did not. And here we are collecting the runoff coming out of each of those soil columns. So again, this is the concentrations of metals and total pHs in those samples. And again, the red is for the test that I'm describing right now. And I just am showing this again to highlight that this storm was not a particularly unusual storm. People from this area will know that last year in September it didn't rain except for, half hour one day, and this in fact was that rain event, but we had to dilute it a little bit with some of our clean runoff on site. So it ended up not being a crazy toxic storm. It was, fairly normal compared to chemical constituents from the other storms. All right, effectiveness. Chemistry side, I'm showing just 2 slides. This one's for metals, and this is percent reduction in the dissolved metal concentrations entering those columns versus coming out of those columns. And we're seeing huge reductions in zinc, cadmium, and lead, and also pretty remarkable reductions in dissolved copper as well. So this, from this perspective, bioretention was an effective tool. The PAHs also showed really significant reductions. So on the left of each one is the untreated runoff, and then the 2 treated runoffs, and then our control water that we used for the fish exposures. So first of all, we did lose pHs to the big plastic mixing tank, which was not a surprise because pHs are hydrophobic, attracted to plastics. And we ended up losing about 58% on average of our pHs to that large mixing tank. However, what went through then is what I'm showing up there in the figure. And there was a further loss of 75 to 90% loss of pHs due to the treatment itself with the soil columns. How about the biology? So I'll show you, these are some of these are the 2 target organisms that we used, mayfly nymphs and juvenile coho. And just a little anecdote for how important this mayfly is as a food item for juvenile salmon, are that when I tried to find an image of a real mayfly nymph, of a Baetis nymph, 90% of the images that came up were actually flies. So these are definitely a preferred fish food. All right, and here's the results of that study just for survival, basic survival of these organisms. So in control water, very high survival of all 3 of these test organisms. In the untreated runoff, 100% mortality in most cases, very significant mortality also for the mayfly nymph. And then in the 2 treatments, there was no difference between the columns with or without plants, but both of them, the treated waters, 100% effectively, 100% survival. No difference from the control. So very effective just from this one pretty coarse metric. I'm going to show a couple more of sublethal effects slides. So this is for the Daphnia, Cereodaphnia, reproductive impact. So this is a test that lasts 7 days while the Daphnia grow up to reproductive age and then produce 3 broods. So you can say they started to produce broods on day 5. And I'm not showing the untreated runoff because they were all dead before they had a chance to start reproducing. So this is just to demonstrate whether or not there were sublethal impacts on reproduction for the treated water itself. And at day 5, there was a slight difference in the reproductive ability. But by day 7, they had all caught up. So in fact, no difference between controls and the treated runoff. It prevented any so lethal and sublethal effects for the invertebrates. For the zebrafish larvae that were exposed, I'm showing some sublethal effects, and this is for heart defects in the embryos, and particularly circulatory issues, circulatory stasis we saw in the fish in the untreated runoff. This is where there's no blood moving through the peripheral system. And also atrial regurgitation, where the heart valves are not closing off fully with each pump through the chambers. And those effects on a statistical level disappeared when the runoff was treated by the soil columns. And this might be the last one here, where I'm showing other sublethal effects for the zebrafish. Basically, at the top, I'm showing that fish exposed to untreated runoff were shorter. They had the pericardial edema, so heart problems, and also significantly small eyes, so that microphthalmia. When the runoff was treated by the soil columns, normal size, normal hearts, and pretty much normal eyes. This is a very slight but significant decrease in eye size, but very significant improvement from the untreated runoff. So as a summary of the various tests that we did to look at effectiveness to date, because we have a bunch of genetic material that we're still in the process of examining, basically we're seeing an elimination of lethal and sublethal responses for pretty much, with the exception of a very tiny impact still on that eye size, we're seeing complete removal of all the lethal and sublethal impacts of the stormwater runoff by passing it through soil columns. And I think that's all the time we have today. I'd love to talk about it more if people want to afterwards. And it looks like hopefully we still have time for questions.